Imagine a world where life is not born but built, where scientists construct living organisms from non-living molecules, encoding behavior, growth, and even self-replication. This is no longer science fiction. The quest to create synthetic life from scratch is one of the boldest scientific frontiers of the 21st century, and it raises fundamental questions. How close are we to achieving it? What defines life, and what is the mysterious Golem Project? Let us begin with the basics. Synthetic life refers to engineered organisms that are either completely artificial or constructed using a minimal set of biological parts not found in nature. Unlike genetically modified organisms, which are altered versions of natural life, synthetic life starts from the ground up, often using custom-designed DNA, proteins, or even alternative chemical systems. One of the biggest breakthroughs came in 2010 when Craig Venter's team announced the creation of the first synthetic cell, called JCVI Sen1. They synthesized a bacterial genome, over 1 million base pairs of DNA, and inserted it into a host cell. That cell then came alive and began replicating under the control of the synthetic genome. For the first time, a living organism had no ancestor. It was powered by human-written code. But how close is this to true artificial life? JCVI Sen1 still used a natural cell shell as a chassis. The genome was synthetic, but the machinery, the ribosomes, membranes, and enzymes was borrowed from biology. This is a major step, but it's more like rewriting the software while using the old hardware. Now enter the Golem Project, not a fantasy creature, but a real research initiative exploring how lifelike behaviors can emerge from artificial evolution. Golem stands for Genetically Organized Lifelike Electromechanics, and it was launched in the 1990s by Hod Lipson and Jordan Pollock at Brandeis University. Their goal: to evolve physical robots automatically in simulation, fabricate them, and watch them move and adapt, mimicking evolution in machines. Ask yourself, what is the difference between a machine that adapts and a cell that evolves? The Golem Project showed that evolutionary algorithms could design novel, functional machines, some with locomotion strategies never seen in nature. Although these are not alive in the biological sense, they reveal a key principle: lifelike behaviors, adaptation, selection, growth, can emerge in artificial systems, whether silicon-based or biochemical. How far can we push this? Researchers are now combining synthetic biology and AI-guided evolution to design protocells, minimal systems that mimic living cells using lipid membranes, DNA circuits, and energy gradients. Some labs are even experimenting with non-carbon-based molecules, such as xenonucleic acids, XNAs, that do not exist in nature but can replicate and mutate. These could one day form the basis for life too, a completely alien biology designed on Earth. Let us pause here and ask, what would it take to build life from non-living components? At a minimum, synthetic life requires a boundary, like a membrane, to separate it from the environment, metabolism to harness energy and maintain structure, information storage, like DNA or RNA for reproduction, evolutionary capacity. It must be able to adapt and change. Projects like Protocell assembly at the University of Tokyo and bottom-up biology at the Max Planck Institute are building these features piece by piece using artificial vesicles, programmable enzymes, and minimal genomes. But is this really life? The answer depends on how we define it. If life means self-maintaining evolving systems with autonomy and purpose, then synthetic biology is getting very close. If it must also include consciousness, intent, or open-ended creativity, we may be centuries away. Could synthetic life be dangerous? Yes, and this is a core concern. If we build organisms that evolve outside our control, we might face biosecurity risks, ecological disruptions, or ethical dilemmas about artificial sentience. That's why many leading synthetic biologists are now calling for safety locks, like kill switches and traceable genetic tags to prevent runaway replication. In a way, the race to build artificial life is also a mirror for our own origins. By trying to recreate the conditions of early life, using hydrothermal vents, electric discharges, or clay mineral templates, we may finally learn how life began on Earth. So how close are we? We have already created minimal cells that can live and replicate, and protocells that mimic aspects of metabolism and behavior. Evolutionary algorithms are designing robotic systems with lifelike adaptability. The next step is integration, combining these elements into a truly autonomous, evolving system that did not arise from any natural organism. It may take a few more decades or longer, but the foundations have already been laid. Would you like a comparative timeline of major synthetic life milestones or an explanation of how protocells differ from engineered bacteria like E. coli? Synthetic life refers to the creation of life forms that are not naturally evolved but engineered using artificial or chemically synthesized components. It includes bottom-up approaches that build cells from non-living molecules and top-down methods that minimize natural organisms to their essential genetic core. True synthetic life must demonstrate autonomous growth, replication, and metabolic activity, should also interact with its environment and evolve over time. This goal lies at the intersection of biology, chemistry, nanotechnology, and information theory. The J. Craig Venter Institute synthesized a bacterial genome, Mycoplasma mycoides, and transplanted it into a DNA-free cell to create a replicating synthetic organism. Creating synthetic life is like assembling a working smartphone from raw silicon, carbon, and polymers. Every part must be precisely engineered and integrated. Golem Project, genetically organized lifelike electromechanics, began in the late 1990s as an effort to evolve robotic life forms using computer simulations and 3D printing. It aimed to create self-replicating machines using evolutionary algorithms and physical embodiment. Though not creating life in the biological sense, Golem pioneered ideas of artificial morphogenesis and machine evolution. The project laid conceptual groundwork for integrating computation, material science, and self-assembly toward lifelike systems. It demonstrated that lifelike behavior can emerge from iterative variation and selection. Carl Sims and Hot Lipson showed how virtual creatures evolved locomotion strategies using only mutation, selection, and embodiment. Glome's approach is like breeding virtual organisms in a computer terrarium, watching evolution unfold in silico before bringing it into the real world. Efforts to create minimal life focus on identifying the smallest set of genes needed for autonomous life. JCVI Syn3 is a breakthrough, a synthetic cell with only 473 genes, the smallest genome known to support independent replication. These projects aim to understand what functions are truly essential, shedding light on the boundary between life and non-life. By reducing complexity, scientists can build controllable, modular chassis for synthetic biology. However, even minimal genomes still rely on a natural cytoplasmic environment to function. JCVI Syn3 can reproduce and maintain viability, but its behavior is erratic due to missing genes for robust regulation. A minimal genome cell is like a stripped-down car engine. It can run, but not smoothly without the full dashboard and control systems. Bottom-up approaches aim to assemble life from non-living chemical components, lipids, proteins, nucleic acids, and energy sources, without using any biological template. Protocells are one key focus. Vesicle-like structures that mimic membranes and can encapsulate biochemical reactions. Researchers strive to get these protocells to grow, divide, and perform basic metabolism. This strategy probes the origin of life and tests whether life can emerge under controlled lab conditions. Key challenges include integration of energy conversion, information replication, and error correction. Synthetic vesicles containing ribozymes have been made to catalyze RNA replication inside lipid membranes. Building a synthetic cell from scratch is like crafting a self-cooking, self-replicating kitchen from individual spices, pans, and recipes. Xenobiology explores life built on alternative chemistries, not just synthetic genes, but alien backbones, new amino acids, or altered metabolic systems. The goal is to expand the chemical space of life and create organisms resistant to natural viruses or uncontrollable evolution. One line of work involves synthetic DNA with extra base pairs beyond A, T, C, and G. Another uses mirror image molecules eGNA instead of ddNA to prevent natural degradation. These innovations offer potential for novel therapeutics and biosecurity. The Hachimoji DNA project demonstrated stable DNA with 8 bases, showing alternative genetic systems can encode information. Xenobiology is like composing music with an entirely new musical scale. It sounds unfamiliar, but still carries structured meaning. Some synthetic life research focuses on digital organisms, software-based entities that replicate, mutate, and evolve in artificial environments. Projects like Avita simulate evolution in computers, where digital genomes encode behaviors and replication rules. Evolutionary robotics builds physical machines that adapt their structure and behavior over generations, mimicking Darwinian evolution. These approaches blur the line between algorithmic intelligence and biological-like adaptation. While not chemically alive, they test principles that underpin living systems. Avita organisms have evolved complex features like logic gate computation and adaptive responses to virtual environments. Digital life is like cultivating virtual bacteria in a computer petri dish. They mutate, compete, and evolve without ever being made of cells. Creating artificial life raises deep ethical and philosophical issues about our role as creators. What rights, if any, would synthetic organisms have? How do we contain or control systems capable of evolving independently? Could life built from scratch challenge definitions of consciousness or self-awareness? These questions drive ongoing policy debates, bioethics frameworks, and public concern about artificial biology's dual-use potential. DARPA and other agencies fund research into safe-by-design synthetic biology to prevent unintended consequences. Making synthetic life is like opening Pandora's box, powerful, transformative, but fraught with uncertainty and responsibility. Despite major advances, no fully synthetic organism, built entirely from non-living parts and functioning autonomously, exists yet. Synthetic genomes still require biological scaffolds, and bottom-up protocells lack sufficient complexity. Integration of replication, energy metabolism, and error correction remains a bottleneck. However, the field is rapidly advancing through AI-assisted design, microfluidics, and programmable chemistry. In 10-30 years, partial synthetic life forms with tailored functions may become widespread in research and medicine. Artificial minimal cells are already being used to study drug delivery, biosensing, and gene circuit behavior. We are at the stage of building Lego prototypes of life. We see the shape, but the engine is still under development. We are edging toward the synthesis of artificial life, but still rely on hybrid systems, mixing natural and synthetic parts. A fully artificial life form built from scratch and capable of independent evolution remains a formidable challenge. Success will likely come from converging fields: synthetic biology, AI, quantum chemistry, and origin of life studies. The implications are vast, from artificial ecosystems to new forms of intelligence and biosafety protocols. Humanity is not far from transforming biology from a descriptive to a creative science. Synthetic life platforms like OpenTrons, BioX, and Venter's Genomic Foundry are pushing toward automated life engineering. We are standing at the edge of a new genesis, not with divine fire, but with code, molecules, and design.